Well, welcome to this seminar of the UMI group on Modellistica Socio-Epidemiologica. And I'm very happy to introduce Michele Piana. Michele Piana is full professor of numerical analysis from the University of Genoa, where he, he leads also the MIDA group on methods on image and data analysis. He also leads the Life Science Computational Lab at the Polyclinic Hospital San Martino, always in Genoa, and he is also associated with the Osservatorio Astrofisico in Torino. Well, I would say that Michele definitely has a very wide range of interests in applied mathematics in the broadest sense, with applications going from computational medicine to solar physics. He has led and still leads several projects, also at the international level. And there's a very dense network of collaborations in and out of academia. So today he will tell us about his mathematical journey into cancer, and I'm very glad to welcome him to this series of seminars. So please, Michele, thank you very much.
I thank you, Dimitri, for your lab, which is that is and the hospital, two focus, one is on neurology and one is on in oncology. And today I will mainly discuss this second topic. Okay. Let me first thank you. A lot of people that that are doing the main part of the research. Most of them are very young scientists and they are very active, and I'm very happy to have them in the group. And let me also thank some financial support coming essentially from research grants that are supporting our research to hire research scientists, young research scientists inside the group. Okay, so as you perfectly know, we are in the middle of a big storm related to use of computational methods in many different areas, and in particular in the case of medicine and of biomedical scientists in a broad sense. So if you talk with medical doctors, most of them since 3, 4, 5 years ago thought that mathematics in medicine essentially means statistics in medicine, or biostatistics, and now things are a little bit changing, very fast changing. And we can say that the main difference between the original way to make mathematics inside research hospital and current way is that statistics in biomedicine is essentially related to study the trends that are concerned with an average view of the patient. While the current focus is about the study of a personalized way of making a computation in medicine. And so everything is focused on individuals and on the investigation of data-driven biomarkers that are related to individuals. I think it's important to point out that, however, the old and the new ways to consider mathematics in biomedicine have some aspects in common. For example, in both cases, what we do at the end is to compute probability distributions. The second thing is that there is a lot of emphasis on the statistical properties of the noise affecting the data. And in all cases, we need to provide medical doctors with an uncertainty quantification of the outcomes of our algorithms. At the end, what I like to mention every time is this quote by Herbert Simon, who is not a mathematician, he is not a medical doctor, he's an economist. He won the Nobel Prize in Economy at I think, 1998. And I think that this sentence is beautiful to understand what's going on now in the hospital. That is, we have a huge amount of data. Let's say we are going to have a efficient focus on the extracted amount of data. Need a which in the case of mathematics essentially means, in my view, that we need to look for model data attention and data analysis of the model.
Michele?
Michele, ci senti?
C'è qualche problema con la,
Sì.
con la banda?
Okay.
Con la connessione,
Sì,
forse se chiudi la camera...
No. Ok, chiudo la camera, ma probabilmente si era staccato il video, scusate.
Sì.
Qualcuno anche ha attivato la trascrizione adesso.
Ok, mi sentite ora?
Sì, grazie.
E vedete le slide anche?
No.
Vedete le slide?
No, non sono più condivise.
Allora è saltato qualcosa. Arrivo,
Ok.
Grazie a voi. Okay, so these are essentially the main topics I will going to talk about today. As you can see, there are different scales and different modalities and different kinds of mathematics that I will use. And the goal, essentially, every time is to search correlations between different kind of data. And these correlations are related to the different scales that I will consider. So, for example, I will start from a coarse scale, which is related to morphological imaging and functional imaging, where I will look for a correlation between imaging patterns and clinical condition. This will be done essentially by means of pattern recognition methods. And the scale is the one of the organ or of the biological tissue. Then I will go a little bit deeper. I will use the dynamical functional data, in particular positron emission tomography data, in order to compute correlations between the ability of the image to represent the signal related to radioactive tracer and the effectiveness of glucose metabolism, in particular in the case of cancer. And in this case, the mathematical methodology that will be considered is related to inverse provenance theory. And finally, I will go deeper inside the cell, and I will consider the correlation between mutations and the concentration of proteins inside the cytoplasma, and this will be done by studying a dynamical system using optimization methods and using dynamical system theory. Okay, let's start with large-scale biomarkers, and this is an image of a vertebra containing bone marrow inside a body and a patient, and this image is recorded by means of X-ray computed tomography. And the idea is to segment, automatically identify the region of the bone where the bone marrow lives, which is the whole of the vertebra, which its technical term is a spongiosa. And this can be done in many different ways. We investigated two different ways. One is to compute the gradient of the image, because we know that the density of the tissue is very high in correspondence of the bone, of the compact bone, and it is much smaller in the case of the inner part of the bone, which is the spongiosa. And so you can compute the edge of the image by computing the high values of the derivative in the image. This is one possibility to automatically determine the shape of the profile of the vertebra, both the exterior and internal profile of the vertebra. But another way is using a generalized version of a very classical visual computation algorithm, which is the half transform. And the idea of the half transform in the original version is that if you look at a straight line in the image space, and you take one point in each in the same straight line, and then you go to the parameter space looking at the straight line equation, for each point in the image space, you have a straight line in parameter space. And if you go to another point in the straight line in the image space, then it corresponds to another straight line in the parameter space. And if you consider many different points in the straight line in the image space, then you have many different straight lines in the parameter space, and all these straight lines meet at the same point, and this point in the parameter space allows you to recognize in an automatic way the straight line in the image space. Okay, what we proved is that this approach can be generalized to a huge class of algebraic curves, and so you can automatically recognize shapes in the image using this approach. And in this way, you can construct binary masks that allow you to highlight the compact bone and the spongiosa region inside the skeleton. So this is the kind of result you obtain. So this glass man or glass woman is essentially the result of this kind of pattern recognition analysis, and we applied this algorithm in the case of a kind of leukemia, which is very famous these days. This is the same kind of disease that is affecting Silvio Berlusconi, which is an advanced chronic lymphocytic leukemia. And what we could prove using this kind of algorithms is that patients that have a great expansion in the spongiosa, in the case of the arms and the legs, so in the appendicular bones, have a higher risk to die due to this kind of disease. Okay, you can apply this approach also to extract the bone marrow metabolism inside the spongiosa in order to provide an analysis of the ability of the bone marrow to uptake glucose, radioactive glucose, starting from the viewpoint that the metabolism of glucose is related to the aggressiveness of cancer. So the idea is that you can combine the CT data, so the morphological data coming from X-ray computed tomography with positron emission tomography. In the case of positron emission tomography, the patient is injected with a radioactive glucose, that is inside, it enters the circle of the blood, and it is accumulated in the tissue where the need of glucose is particularly intense. And this is typical of tumor, since tumor is full of blood and needs a lot of energy to replicate its cells. And so there is a big accumulation of a radioactive tumor, of radioactive glucose inside the tumor tissue. So what we did is to use this approach in order to determine whether a leukemic patient that underwent a bone marrow transplant is able to reconstruct the bone marrow activity as if it were completely healthy. And this is what we obtained here. You can see a control patient here, which is an old guy who has a different disease with respect to leukemia. And then this is what happens to two patients. One is a leukemic patient that has been transplanted using cord blood tissue. And this is another leukemic patient that has been transplanted using a autologous transplantation. And as you can see, you have the fact that the active bone marrow cells begin to populate regions that were completely inactive in the healthy case. And this is a very strange behavior that is typical of children, of young people. If you look at the bone marrow of young people, you can see that they have active bone marrow in the legs. And in some sense, it seems that the transplantation is rejuvenating the patient, and it's making them go back to their infancy age. Okay, so this is what happens if you consider large-scale biomarker associated to imaging, and applications that we are considering now. What we are trying to do is to look for biomarkers that are related to the skeletal muscle. There are models that are theoretical models that are investigating the relation at an enzymatic level between the tissue in the skeletal muscle and the clinical behavior of leukemic and in general cancer disease. This is an application that has been considered of prostate cancer. And in this case, we used a partial differential equation approach to segment a specific kind of skeletal muscle, which is the red here. And we could prove is that people have a low density in the X-ray tomography signal and a high uptake of radioactive tracer in positron emission tomography signal have a higher probability to have metastasis with respect to people that have a high density in the X-ray tomography signal and a low signal in positron emission tomography. Okay, so a very old-fashioned approach of the search for biomarker is called radiomics. The idea of radiomics is essentially one to correlate the genetic profile with respect to shape of oncological lesion, of tumor lesions in images, mainly in X-ray tomography and magnetic resonance imaging. A typical radiomics workflow is the following one. So you first make an image acquisition. In this case, you have a brain magnetic resonance image acquisition. And then that is able to automatically highlight, point out the presence of a brain tumor. For example, this is a meningioma in the image. So this brain tumor is extracted, and then you apply algorithms that are able to compute a lot of number, namely a lot, so typically hundreds of number that are related to the morphological, geometrical properties of the highlighted, of the segmented lesion. And then you can do essentially two things. You can stratify, that is, you can cluster the patients in this very highly dimensional space of hundreds of numbers using unsupervised machine learning, or you can train neural networks in order to predict the follow-up of the disease using these numbers extracted from the lesion. There are several technical aspects that we are considering now. For example, we are trying to compute scores related to the reliability of the features that are extracted by using this feature computation algorithm. And we are studying different ways to make a complexity reduction. This complexity reduction issue is related to the fact that the number that radiomics code extract from the lesion are too big and they are redundant. And so the point is to try to look for correlations that decrease the number of significant features, where significant means the ability of specific feature to be actually really predictive for the disease follow-up. And this is, again, a very technical and open issue in this field. And then we are trying to apply different unsupervised machine learning approaches for clustering. And what we are realizing is that you need non-linear approaches to make an effective clustering of the radiomic features. This is an example in the case of multiple myeloma, which is another blood cancer disease. Of course, we need to provide medical doctors with computational pipeline, and this is an example of the pipeline we have implemented in the hospital. And as you can see, this is the kind of numbers that you have to extract from the lesion. What we are doing now is to validate, in the case of meningioma, is to validate the pipeline using biological and biochemical biomarkers in order to understand whether the image-driven biomarkers are compatible and as reliable as the biological ones. And the other thing is to try to implement deep learning approach. That means convolutional neural networks that utilizes as input the images instead of utilizes as input the extracted features. In this kind of networks, the algorithm is able to automatically extract the features and to make the prediction of the disease outcome. Okay. Then going deeper in the analysis, so we go from the tissue, which is represented in the images. We go down to the cell, and the main application we are considering now is the construction of in silico models of cancer cell in specific in specific period of its maturation during during its life. In particular, we focused on colorectal cancer, and what we did is to construct a model of the chemical reaction network, a mathematical model of the chemical reaction network of the colorectal cancer cell in its G1 phase, which is the cellular phase just before mitosis, which is the cellular phase where the division of DNA begins. This cancer cell we considered in this big network, which is essentially a graph. All the color boxes are represent a protein inside the cytoplasma. This gray barrier here is the cellular membrane, and all the black arrows are chemical reactions that involve the different proteins inside the cell. So our chemical reaction network, so the chemical reaction network we considered for the colorectal cancer cell is made of around 10 pathways, which means that overall we had to consider more than 400 proteins and more than 800 chemical reactions. Each chemical reaction is a differential equation. So at the end, we had to solve a big Cauchy problem, a big dynamical system, where the idea is that the equilibrium is the idea is that the asymptotic solution of the dynamical system represents the equilibrium of the proteomic profile. More specifically, in this system, X is the vector of the concentration of all the proteins in the network, S is the stoichiometric matrix, and V is the vector fluxes that connect the protein concentration with the kinetics constants K. Of course, we need an initial condition here. We have information about the K vectors from the literature, the scientific literature, and what we have to do is to compute the equilibrium at big times, because this equilibrium represents the concentration equilibrium for the proteins inside the cell. The next thing here is that you can modify the dynamical system in order to implement the presence of mutations inside the DNA. So what happens is that a cancer is essentially a collection of mutations. The number of mutations that is rather small. In the case of cancer, colorectal cancer, the number of mutations is around 10, while for liquid cancers, you have even less mutations. 2 or 3 mutations are really significant for the cancer to occur. The nice thing here in this model is that each mutation can be implemented inside the dynamical system in 2 different ways. The function mutations are implemented by modifying the shape of the stoichiometric matrix, while the so-called loss of function mutation are implemented by changing the initial condition. So what you can see here is what happens to the proteomic profile of a colorectal cell when you implement a specific very significant mutation. This is a gain of function mutation that is typical of colorectal cancer. And in the x-axis, you have all the proteins. The black dashed line is the healthy situation, while the red one, the red solid line, represents the diseased condition. So in this way, you can do many different things. For example, you can understand how the different mutations combine in changing the proteomic profile, or you can compute whether the order with which the mutation occurs has some impact on the differences in the variation of the proteomic profile at equilibrium. The main application we are considering now, this is a project that has been funded by Roche, is to understand in silico, so in using this computational model, which is the impact of different biomedical chemotherapy drug, both the ones that are already considered as clinical tools to address this kind of cancer, but also possible new molecules that can be considered for chemotherapeutic effects. The nice thing is that in this model, you can also implement the amount of drug. You can quantify the amount of drug you can consider as the optimal amount for making the chemotherapy effect maximum. And you can also investigate the effect, the impact of the use of a cocktail of different chemotherapeutic drugs in a very quantitative way. And this is something that we are doing now together with Roche technicians. Okay, so we can go deeper in the cell, but first let me tell you something more about what we are going here. One of the important things is how to construct this kind of chemical reaction network. In order to construct this network, we worked for almost 2 years together with oncologists in the hospital and together with people from Roche. And essentially, the approach we used was essentially to read papers in PubMed and to look for the proteins involved in colorectal cancer and the kind of reactions involved in colorectal cancer. But now we have machine learning, and in particular, we have natural language processing. And so what we are doing is to construct a neural network that utilizes approaches typical of NLP in order to automatically construct these kind of networks using PDF from the literature. And in this way, our objective is to construct an automatic pipeline that is able to generate at least a first prototype of chemical reaction network in silico for any kind of cancer. And for example, what we are doing now is to do this in order to construct a chemical reaction network for 2 different kinds of leukemia. Then the other thing is we are working together with physicists in the hospital that are measuring the proteomic profile in cancer cells and are able to measure the kinetics constants K in the different kind of cancers. So in other terms, what we are doing here in these equations is to have measures of the equilibrium of the proteomic profile using mass spectrometry, and we are trying to solve the nonlinear inverse problem that relate the equilibrium concentration to the K in order to have a measurement-driven estimate of the K instead of using the ones coming from the literature. And finally, what we are doing is to try to speed up the computation of the equilibrium. Instead of looking for the numerical solution of the ordinary differential equations. What we are doing is to transform the dynamical system into an optimization problem, and we have implemented a ad hoc numerical optimization algorithm for the computation of the equilibrium that is speeding up a lot the computation, and also it's more accurate than the use of standard methods for the numerical solution of ordinary differential equations. Okay, now we are inside the cell, and once we are inside the cells, what we want to do is to go inside the nucleus of the cell and to look for mutation of the DNA. And what we are doing here is to construct pipelines, computational pipelines that are bioinformatic pipelines in order to extract mutations and compute mutations from measurements of the DNA and of the RNA and of the exome and so on. In the hospital, we have many different kind of devices for making these kind of measurements. The bad thing is that these measurements are affected by a lot of noise and by a lot of artifacts. And all the mathematical game here is to get rid of the many ambiguities that are related to these kind of measurements. Also, what we need to do is to be as fast as we can with this kind of computation, because the medical doctors are using this kind of data more and more frequently, and so we are accumulating a huge amount of data that needs to be analyzed. Besides the technological aspects that we are trying to address, there are two mathematical problems that we are studying. One is related to the fact that these pipelines for the analysis of DNA data are often provide ambiguous solution. For example, different configurations of the outcome of the algorithm are compatible with the same situation in the DNA, and so we need to make a model selection at the end of the bioinformatic analysis of this kind of pipelines. And this can be done by using many different approaches. At the moment, since we have a lot of computational power in the lab at disposal, we are using very coarse grid search algorithm, but we need to look for something more refined and more sophisticated in order to be more effective in this analysis. And finally, the other problem we are considering at the moment is the use of multimodal data and our attempt to put together many different, completely different kinds of data that we have at disposal, in particular, in this case, for the study of lung cancer. We have to combine data that comes from DNA, from RNA, from proteomics, and from clinical records. And in order to do this, we are essentially implementing methods that use Bayesian approaches and sequential Monte Carlo approaches. Okay, so this is essentially what I would like to say for today. Let me also say that we are hiring people. We are desperately looking for young scientists that can come to Genova working with us in the hospital and in the lab in the master department. And so if you are interested to this position, please have a look at our webpage. Okay, thank you, and I'm ready for possible questions.
Thank you very much, Michele, for the very interesting contribution and very ample in contents and arguments. So is there any question from the audience? Please feel free to ask. Okay, now we can see you again.
Maybe, I don't know if I have to write it or I can ask.
No, you can ask Ezio. We can hear you.
No, I was just wondering because, Michele, you have told us that you are modeling about 850 chemical reactions. I wonder, are the parameters known from the literature, or I mean, how do you deal with the parameters in those
Yes.
equations?
That's the main problem at the moment. Let me go back to that slide. Let's see, 1 second. So if you look at the dynamical system, that I'm sure this kind of dynamical system you know very well. So in this dynamical system, the data we know are the initial condition, and this comes from the literature, and the K that comes from the literature. Then we have the information concerning the stoichiometry matrix, and we have the information concerning the fluxes. And what we need to compute is the equilibrium, so the asymptotic solution of the problem. But what we want to do is to get rid of literature, essentially, or to try to use less literature. And this can be done in 2 ways. For example, what we are doing now, we can measure the concentration profile at equilibrium using mass spectrometry. So once we measure this equilibrium, which is essentially a asymptotic solution of the dynamical system, we need to solve the nonlinear inverse problem to determine the K from experimental measurements. Once we have the K, then we can use the model and the computational method for computing the equilibrium to make all the tests concerning mutations and chemical drugs. Okay?
Okay, thanks.
Okay, okay.
Other questions? Please feel free to advance.
May I?
Sure, Fasman.
Okay. Thank you for the nice talk, and just if you can say something more in more details about the technique that you used in order to avoid to compute the equilibria and instead using an optimization
Okay.
technology or approach. I don't know.
Okay.
Moreover, another question.
Yes.
If you have, I see that you have a stoichiometric matrix. I'm wondering if it's not a full rank matrix, and if some linear invariants are preserved, and in this case, if the numerical techniques that you used take into account the preservation of linear invariants.
Okay, you are asking me a lot of technical things, and I'm not sure I will be very. So I
Sorry.
No, okay.
Because it's interesting.
Most of my time is to try to make people work instead of working, but I will try to answer. Okay, let me show you one paper that we are writing now. Actually, we have already submitted it, and so can you see the paper? So what you can do, let me see if I find the equation that I'm looking for. What you can do is that you can prove that the dynamical system I showed you before is equivalent. Let me see if I see the equation. By the way, I've not written I'm not involved in this. Okay, you can prove, you can use this conservation matrix here, and you can consider a weakly elemented assumption for the dynamical system. And what you can prove is that the dynamical system is equivalent to a zero finding problem, where the function you have to of which you need to find zero is this one, okay? Where this is a part of factorization of the stoichiometric matrix. And this is the matrix that you construct using the conservation vectors. Okay?
Yes, I see, I see.
Okay, so what you can prove is that if you look for the zero of this, you find the equilibrium solution.
Yes.
And then what we did is to look for numerical, essentially gradient-based, not just gradient-based, because with gradient-based, simple gradient-based method, it doesn't work so well, but something using gradient-based method, you can compute the zeros of this equation, and you can find the equilibrium.
Okay.
Okay.
Thank you. Very nice.
Second thing, the stoichiometric matrix is rather sparse. It is structured. You can use, again, conservation laws in order to study the properties of the matrix, of the stoichiometric matrix. And this is something that we are investigating now.
Okay, so I can send you something we have done.
Oh, I would be more than happy. Yes.
Okay, thank you.
Okay.
Okay, there is actually a question from Michele in the chat. I think you can read it as well, Michele. From another Michele.
From another Michele, let me look
Yes.
for the chat.
He's asking about some details on the...
I'm not seeing it.
Okay, he's
okay.
asking you if you can give some more details about the analysis of the functional data you mentioned at the beginning of the slide where you were talking about chasing for correlations.
Okay, so I think in the imaging data.
It was in the first part.
Okay, so essentially we are considering two different kinds of images. One are structural images and the other one are functional images. So structurally, in this case, for example, you can see what we do in the case of X-ray tomography imaging, which is a morphological structural imaging modality. And what we are looking for is correlation between the pattern in the images and the behavior of the disease. And for example, what we could prove here is that if you look at the whole of the bone and you consider you are able to measure the expansion of the hole in the bone, what we can prove is that people that have a notable expansion in the appendicular bones, in the whole of the appendicular bones, have a greater probability of dying for chronic leukemia. This is the first thing we proved and we published. The other thing is you can use functional data, for example, positron emission tomography data. In this case, you take a glucose, you make the glucose become radioactive, you inject the radioactive glucose inside the person, inside the blood, and you use you exploit the fact that the tumor is very hungry of glucose, and so it wants to eat as much as glucose it can. And it is so stupid that it is not able to distinguish between normal glucose and radioactive glucose. And so once the tumor accumulates radioactive glucose, it begins to emit lights. And so you can measure the aggressiveness of the tumor as a correlation between the amount of light it emits. Okay? And we use this approach, for example, to evaluate the effectiveness of bone marrow transplant in the case of leukemia patients, and to understand what happens to the skeletal muscle of people with cancer, like, for example, in the case of prostate cancer, because there are some biological models that are saying that there is a correlation between the properties of the skeletal muscle and the aggressiveness of the tumor. And what we could prove is that there is a correlation between the aggressiveness of the tumor and the density of the skeletal muscle and the avidity of the skeletal muscle to eat radioactive glucose. Okay, so these are the kind of correlations we are considering. I cannot hear you, Dimitri. I cannot hear you. Dimitri, I cannot hear you. I cannot hear you.
Dimitri, you're muted.
Yeah. I was mute. Sorry. No, I was saying that the other Michele was thanking you, and he just commented.
Okay, yes, I now I can read it. Okay,
you can read it. So you can read that he says it's a really interesting lecture, and of course I agree with him. I don't know if there are any other questions from the audience. Otherwise, I would like to share a comment and maybe ask a very general, but I realize it's surely a naive question, but you have a lot of experience. And of course, this is the era of artificial intelligence, machine learning, and all that kind of stuff. And
it's a kind of speculative bubble.
Yeah, so. Sure, but many scientists are using these techniques, and of course, we have mathematicians approaching this stuff, physicists, engineers, economists, and whatsoever, also sociologists. And all they have the level of knowledge about these tools, and also their level of trust, maybe. So since you are in contact with many medical doctors, I would like to ask you, what is your impression about their level of trust in this kind of...
They are extremely suspicious. Mm-hmm. And they, let me see, my opinion is that they are completely right to be very suspicious. Because if you ask clinicians all over the world if they are now really using in a systematic way artificial intelligence techniques to make prognosis on diagnosis or to support the therapeutic intervention, you will find very few applications where this happens. This is due to many different things. One thing is that it's not true that we have, in my opinion, that we have so many data. For example, we do not have so many genetic data at the moment at disposal of data. I'm sure that we are going to have a huge amount, a huge increase of this data. But at the moment, now in the hospital, we are not sequencing so many DNAs, okay? And you know that you need a lot of data to train neural networks to make them trustable. Yes. So this is the first problem. At the beginning of the COVID, I was in Houston at a very big conference in medical imaging, and there was one afternoon where radiologists collect all the mathematicians and computer science in one room and began to ask, okay, you did this, I tried with my data, and it didn't work. So please tell me why. Okay, it was very frightening, okay? So I think we need to work much more than what we are doing now in order to be really, really reliable for medical doctors. Another thing, I really think that AI is not just machine learning. AI is much more than that. So it's inverse problems, it's optimization, it's dynamical systems, it's a lot of things. So AI is, machine learning is essentially purely data-driven. Things are changing, but the old style was purely data-driven. I think that we need to plug in the algorithm also information on the model, on the mathematical model that come from biology.
So a sort of combination.
Of combination of the
first principles, modeling, and
yes. So a kind of model-driven AI is probably what we should do at the moment.
Okay. thank you very much for sharing your
Okay.
experience and opinion.
Okay,
And thanks,
thank
of course, for the contribution.
Okay.
So if there are no other questions, I would thank our speaker again.
Thank you, Dimitri, and thank you, and happy Easter.
And of course, you should know it's already advertised in the website that we organize a workshop in Naples next May.
Okay.
So you're really welcome to come.
Okay.
And it's 18, 19 of May.
Okay.
And
okay.
it's the first occasion where our group will meet present.
Okay, good,
after
good.
3 years, I guess.
Okay.
We will send an announcement soon.
Okay. Okay. Thank you. Thank you. Thank you.
Okay.
Okay.
Thank you very much. So
Thank you. Thank you.
thanks to everybody. And
welcome.
see you next.
Bye bye. Bye
Bye.
bye. Bye. Thanks.
Ciao, grazie.
Ciao,
Thank
ciao,
you.
ciao, ciao, ciao,
Grazie. Ciao.
Ciao, ciao, ciao.